So back to 2010, it's about 7 years ago, there's a paper appeared in Nature Review called Phenomics, the Next Challenge. There's a paper talking about a lot of issues. But the message is that there's a long overlooked issue in biology, which is the phenomic characters. And sometimes people thought it would be easy to get characters, but if you think really carefully, you found that it's not so straightforward. So these phenotype characters are something like the health, the disease, or evolutionary patterns, or in other cases could be the weather data and something else. This data could be important because it can help us understand the interactions between the genotype and the environment. But today, our ability to categorize the phenomes is left behind our ability to categorize genomes. There's lots of efforts being spent on next generation, the third generation sequencing. Companies like Sanger Sequence, Illumina, PacBio has spent lots of efforts to sequence the human or other organisms' DNAs. So you can fairly get the DNA of a patient in the about 100 bucks. You can also further study a patient by looking at the epigenetic data, the metabolomic data, the proteomic data. All this can be done in a standard way. So you can fairly get those genome side, data on the genome side, but what about data on the phenome side? It's still not there. So while the biologists see the challenge, the IT companies see this as an opportunity. So those companies have been spend effort to develop these personal wearable devices. And on top of that, there's software being developed, or a framework being developed to analyze the data collected from these personal wearable devices. With that, they hope that these devices can educate and motivate individuals towards better habits and better health. So if you look at the phenomes from the data point of view, there are two subcategories. One is the hardware development. The purpose is to collect the data with is a fairly low cost, stable, and systematic. And from the software side, we want to analyze the data, we want to mine the knowledge out of it, to figure out what does the data really mean. So if you look at the hardware side, we require some devices that's really fully automatic. Doesn't require any manual involvement. It should be high throughput, get thousands or even hundreds of thousands of data in a day rather than just one value. It's a large scale, so you can cover a large population. It's non-invasive, so you can study a subject over time without interfering. It should be done in natural dynamic environment, so you don't really have to force the subject in some situation. For example, a patient must go to hospital to measure the phenotype. So this is some special requirement for this kind of phenome data collection. On the software side, there are lots of raw data of signals being collected from sensors. It includes lots of noise, or sometimes the data is misleading, is biased. We have to process them. And some of the sensors are imaging sensors, so we need to extract features from images before using them. We have more than one type of phenotypes, so we have to model them together. Sometimes we also look at the environment, because the environment is changing. If we only look at the phenotype, we cannot figure out what we are measuring. And eventually, we want to integrate the genome, the DNA sequence, the protein activities into the system so that we can answer the questions such like, we see this phenome is because, under this special environment, a gene mutation will result in certain type of epigenetic pattern, so on and so forth. So, in my previous work, I have worked with some biologists and computer engineering colleagues to develop a phenotype data collection platform. To develop the platform, we spent a lot of time to adjust this hardware setup. In the center here, you can see this is the ray of light. Oh, by the way, the project I study are focused on plants. For plants to grow, we need sunlight, we need water, nutrient, and the soil. So if we want to see how plants grow over different conditions, we need to replicate the change of these factors in a growth chamber. So in the chamber, we have different light zones, different lights. We have a special sensor that can even sense the change of the temperature of the surface. We have developed the control board so that we can control the light zones. So the plants growing in the corner of the box will receive the same amount of light as the plants growing in the center of the box. There's lots of engineering here. So after getting the system down, we tested the hardware in different settings. So this is the growth chamber setting because this is as tall as a refrigerator. Okay. And these are the light beams, the LEDs here. And this person had to climb up a ladder and to operate the computers put on top of it. And this is a closed environment. We put the plants in here and fill the box with carbon dioxide. So it's a very high carbon dioxide concentration. And then we can check how the gas exchange is in this closed environment. Well, this is a more challenging setup. This is greenhouse. We even don't know what's the day is for that day, but still we want to get the phenotypes in the environment and try to figure out the meaning of the data. So these are the setups, and this is one of our lightest chamber. We have about 100 plants growing in it, and you have the light zones here so that the light is extremely even, and they have sensors, so you kind of see, but they're just LED cameras. It's CCD cameras that they can get the fluorescent image. So with this device,
sorry, I already said this. Where are all these plants and things?
where are these?
Are they like on campus?
Not yet. It's the work I have done in my previous place. It's in Eastern Michigan.
Okay.
Yeah, but we distribute it to many different places right now. So you can access it. But of course, it requires lots of investment. But it's doable. Okay. So with this device, we collect lots of data. The data can be divided into two parts, the images and the numbers. We have more than 100 terabytes of images. They are not just your images collected from your phones. These are all from rigorous scientific experiments. And each image includes about 30 to 50 plants. Fluorescence images, and they are systematically captured. That's temporal. They are under a well-controlled environment, and it's dynamic and reproducible. And these images are not just for good looking. So some images are taken under extreme conditions. Very strong sunlight is 10 times sunlight, and if you expose a plant under that sunlight just for 1 minute, it will burn out. So to achieve that high sunlight, we have to use several car batteries to charge an array of flashes. And some images are taken with an ultra-fast shutter speed because we want to capture the activity of the electron flow that changes very quickly from time to time. So the images, you can imagine, very noisy. And some images are taken under totally dark conditions, so the signal is very weak. So this poses lots of challenges in computer vision. On the other side, we get heterogeneous phenotype data. They're not just one kind of phenotype. There are about 10 times different phenotypic parameters. And we know that they are somehow non-linearly correlated with each other, but we don't know how exactly it is. The data size is large, it's about 100 billion images. And these plants have different genome background. Some are mutations, some are wild types, some of them are different cultivars in different places of the world. We replicated the weather data we collect from weather station. For example, in Virginia, we have a weather station. We collect data from there and replicate in the lab. So it's dynamic and reproducible. So this data is also kind of messy. It's because if you test the same plant twice under the same condition, you'll get different things. So it's easy to understand. For example, if I ask you to run for 5 miles and I monitor your heart rate, and after that, don't rest, run another 5 miles. Then I monitor your heart rate, you'll see that the heart rate is totally different. This is just a single example. We have 2 kinds of plants here, the wild type and the mutant. Well, the color represents the photosynthetic activity. The data are collected under in different time of day, under different light conditions. In the morning, you can see the color is different to the color in the moon in the afternoon. You can also see that the wild type and the mutant are different in terms of the changing trend. So by monitoring change trend, you can figure out whether this mutant is doing something wrong because of the missing gene. Anyway, this is just the very rough idea. So in our idea, we think that if you wanted to identify a special phenotype or you wanted to categorize the phenomes, you have to use the knowledge from bioinformatics, understand the genes, the pathways. We need the algorithm in computer vision to study the images, huge amount of images. We also need the data science algorithms to study the numbers to figure out which subject is different to others. So my today's talk is basically here. This is a computer science seminar, so I will omit this part. And we have done lots of works in computer vision, but we can talk about next time. So today we'll talk about the data science. Imagine that we can decipher all the images and turn them into numbers. The next question is, are there anything different from the others? How can we find out? For example, this is the data we collect. There are only 3 phenotypes here. The phi 2, QI, and QE represent different photosynthetic meanings. And this, in each of them is a typological matrix. You have the genotypes in the rows and the time in columns. Okay. And each one of these rows values are much lower than others. Okay. Visually, you can say, but the computationally, one say, okay, we can develop an outlier detection algorithm to find all of this and figure out what's wrong with it. In literature, there are lots of papers, lots of works that have been developed to study this problem. For example, this is a Phenotypic signal 2010. They look at the age of people. This is the age. You see, there are 2 peaks here. So lots of people die very young, this infant, and then there's lots of this another entity. So if the distribution is different in data compared to this the distribution, you can see that the data could be wrong. There are other algorithms developed to tackle the outliers in temporal data. For example, there are lots of peaks here, and this can be smoothed out using the method for the UKY-Computer. But we think maybe this algorithm does not really workable in our case, because we wanted to get the data from weather station and bring it back to the lab. So you can see with the time, the light change really abruptly over time. And when we get the data will collect this. So it changes quite rapidly. So the problem is, how can we tell one of these is different to the other in terms of this color intensity? We also found this problem is more challenging because we wanted to classify 2 kinds of outliers. One is the really wrong values. That should be deleted. These are data from sensors. The sensor may malfunction at some time, so data may not be really useful. But others are biologically interesting to us, something really useful to us. We want to highlight them and bring them to the users. So you can see there are 2 extreme cases. If we put the outlier in the wrong category, maybe we give the user wrong values, or we throw away really interesting results. But the only thing we know is that they are just look different to most of the other data. So how can we differentiate the two types of outliers? I think this could be a common problem in many studies. So we consult biologists to say, how can you tell whether the data is really wrong or it's really interesting? And they said, you can look at the relationship between the environment and the photosynthetic rate. But look at both of them. And there's a model there called MMK that model the relationships between environment and the phenotype data. So we tried it, and we found it really fits very well. So the dots are the data that we collect. These are clean data, by the way. And we found that if we fit this function to it, we get a very high R-square value. That means our model, maybe we can use this model to figure out if the data is wrong. So idea is here. So these dots are the data, and this is the line that fit to this data using the function in the previous slide. So you can see the fitting is not very well. And if we can identify these 2 points, then remove them from the data set, then we get a much better fitting results. So given this just a simple observation, we figure out the optimization problem 1. Want to achieve optimal the curve fitting results by identifying and remove the anomalies. And this will be done on each specific genotype. And if we do this for multiple genotypes, we'll find that some of them will look different to others in terms of the shape of the curve. So this becomes optimization problem 2. We can identify the curve parameters, which here is half-life I1/2 for the half-life. Is most different to the others. So it seems that by using a knowledge-based model, we can answer both questions. So we translate the biological problem into a set of optimization problems. And here are the challenges. The phenotype plasticity is a well-known problem. We have a simple equation to describe the pattern, but it doesn't really hold under all the conditions. Sometimes it holds, sometimes not. It's just a simple equation. And the noise rate could be quite high. And some values are just biased instead of being noise. So to this end, we develop a simple model called the cost to define the process. It's a cost model. We generate candidates of the outliers. And in the refined model, we divide the data into different regions and study the distribution in different regions, and finally put them together. Use the key nearest neighbor model. I won't go to the detail because what I want to do today is to give you the overall idea. So in this defined stage, we use the EM model for the optimization. We assume that the data follow multi-Gaussian distribution. So we use the GMM model here. So here we say, okay, here we have 2 peaks. This left peak probably be the signal because it's very high. Or this peak could be the noise. Let's assume this is the case. And mark this as noise and this as a true signal. So we can divide the data into two groups. One is the normal value and the normal value. So in the E step, we compute the difference between the model and the real value, and adjust the parameters so that the difference is minimized. Okay. After that, we know that how the curve should look like. So we go back to the M model to re-justify the distribution to figure out which one should be moved to here and which one should be moved out. Okay. So this is the refined process. And then we found that our algorithm performance is much better than others. So these are the existing 2 algorithms. This is the first version of our algorithm, and this is the second version. So in terms of the precision report curve, we can achieve a pretty good performance. And here is the score of the MCC. Here we want to test whether the model is sensitive to the chosen parameters. The key parameter here is called the confidence interval. Normally we will say 95% to 99% efficacy. But we want to change it from about 70% to 100%. And you can see that our model is quite stable in this range. it really doesn't matter what the confidence interval threshold you choose. So here we have marked and removed the wrong values. Now how can we find the biological?
In your experiment of this work that we discussed, how many outliers do you find?
Its ratio is low. It's about 0.1%.
But still, there are quite a number of them if you have any.
Yeah, we have 100 million data entries. So yes, total number could be.
So I can mention this initial curve as shown in this picture. Lots of things are far away from this curve. You have to pick a subset.
Yeah.
There are lots of subsets. So how do you handle this issue? Because it seems with the computation it has to be extremely demanding unless something smart or some heuristics is used.
Yeah, we use yes, very good question. We use a temporal window approach. So you have the sliding windows. The window is very decided with the window decided by biology, so we don't know how they choose it. But they usually choose a reasonably wide window. So we don't have too many windows. And the second one, regarding to the computing time, we take a check because then if we find that if one of if one genotype data is wrong, we want the cross check then probably because the sensor is wrong. So if the sensor is wrong, then all the data that's collected by this sensor could be wrong. So we can broadcast the marked errors to the other samples that we haven't tested. So the next step is to figure out our interesting outliers. The challenge here is still the reliability of co-fitting performance. It could be low. Some data may be biased. These are data from our own sensors. It's kind of home-cooked devices, and it fails quite often. So we really need to figure out whether this data is wrong, biased, or we can use it. So we developed a iterative model to find out biological interesting outliers. The idea is that we can divide data into different windows, and some of them the data in the windows they are reliable, and some of them are unreliable. So if they are unreliable, we'll use the data in the reliable windows to help to improve the performance. So this is how we do that. It's not very clear, but I'll try my best. So this is the data set that we collect. So you can see that some of them are wrong here. It's extremely low, shouldn't be so low. And if we fit a curve to this, just the left side of the curve, we found the fitting and match very well with the ground truth, even though it's level is high. But in the bottom, in here, in this part, amplified here, you find that the fitting is around here. Okay. But the ground truth is here. Oh, sorry, the fitting is here. Let me check. To miss some lines here. So this is the fitting results. And this is the results that transferred from neighbors. As we said, if this window is not reliable, we use data from the neighbors to help to generalize this value. And this is our model performance. And this is the final result. So our model is recognized between the ground truth very well, or the other, if you only the local fitting, you have see a quite high deviation. We need some noise fitting. So here is how the model works. So this is much clearer. This is the fit results, and this is our model, and this is data learned from the labeling windows, and this outlier is the ground truth. Our goal is to optimize the parameter such that it fit best with both the local data and the code that we learned from the generalized model from the neighboring windows, so that we can satisfy the result present to the point, the data in this window. So we can eventually need to optimize the probability of the current parameter giving the length blue line and the probability of the data, the points giving the parameter we choose. This is the model performance. We can see that with the window size is 6, our R-square value is 0.97, but if you use traditional methods, it's 0.6. The performance does not hold when we increase the size of windows, but the biologists insist that this is the best window size we want to use. Well, this is the synthetic data. You can see the synthetic data for the other methods. The R value, R-square value is negative, but ours is still very good. So what are the real cases? We found 2 classes of outliers in our data set. One of them keep increasing, another keep decreasing. We asked the biologists to verify them for us, and they found that for the class 2, it looks still normal in terms of the key parameter QI. While for the class 1, the value is significantly lower than others. Then we said, okay, what has happened to this class? And the biologists found that, in fact, in the class 1, this is a active metabolic process, a process called proteolysis. The proteolysis is a process to degrade a major light harvesting protein. So this protein will be degraded to the 2 parts of it. So that the photosynthetic activity will be stopped. You know, the key protein there, if it's missing, then the workflow can't continue. Then we found that in the class 1, we have the overprotection event. That means if we have a little disturb of the environmental conditions, then the class in the class 1 will say, no, we can't move forward, we have to stop, we have to take a break now, we have to rest. While in the class 2, it's like, oh, still okay, you can move forward, it doesn't matter. So we found the 2 different outliers in this data set. So far we talked about the outlier detection, and then the next one we want to discuss is, are there any similar phenotypes in the big data set? It's called the clustering. When we look at our data set, we found that we have to consider. Sorry.
This is a question about 2 slides ago. In this example, the outliers were outliers because they actually were like plants working differently, right?
Yeah.
But before were outliers measurement error?
Yeah, they're measurement.
So do you want to remove plants that act differently in your model, or do you only want to remove measurement errors?
We only remove the measurement errors. We will highlight the biological outliers. So the biologists can look at the verify.
Oh, okay.
There's the 2 extreme cases. But biologists are not comfortable. Said you can't remove anything. You can only mark. So we mark these errors, and then they decide whether the data can be removed or not. So as Merrick said, there are thousands of points here, but they want to go through all of them.
They want to be a problem.
Yeah, right. So here, the second case is, when you look at these parameters, you realize that they are related, and that they have certain relationship, they are in the same complex. And what this is the mimic of these pathways. So one is this, you have the water, you have the pair here, and you have a hole on the bottom. So you can pull the water in, and the water will come out. So the more water you pull in, the more water will come out. There are some related parameters. So in our case, we want to consider multiple phenotypes simultaneously in the clustering process. So as said, we have this matrix. If we translate this to a 3-dimensional figure, it will look like this. So you can see that we can model that data simply use cloud points by ignoring the order of these points. So this is just a 2D display. You can see one is this, one is this. Clearly, they are not Gaussian distributed. The model we developed is to find key optimal density functions from F1 to Fk to better describe the probability of the data given these functions. Which can be done very easily if we assume that the data follow Gaussian distribution. But as you said, as you see in this slide, they are not. So we can do that. So instead, we adopt the non-parametric density estimation approach, which is we randomly choose the landmarkers, L landmarkers, and estimate the distance from every point to the landmarker using kernel function. And then we can pose this S function. And by replacing this part with this S function, we get we can rewrite the likelihood function. Quite straightforward. We use EM process to study the problem. In the E step, we estimate the posterior probability. Yeah. And in the M step, we adjust the membership of the points so we can reconstruct the F functions. We estimate the performance by using some known taxonomy data, the error site, and the ATTED. We compare with the traditional class 3 method. We found our method has better performance compared to the others. So here I introduce you the 3 recent works that we have done in deciphering phenotype data. I would say this is still the starting stage. We haven't considered environment into the model. We haven't considered genotype data into the model. For the data extraction, we use very simple computer vision algorithms. There are lots of hidden information there that they are waiting for us to mine. And in the last slide, I want to share, thank the students that I have and the postdocs helped me to build this platform, and also thank the funding agencies to support the platform. Thank you.